Dr. Noll. I'm a biochemical geneticist. What that means is I am a doctor of biochemistry and genetics, so I have kind of two doctorates. Before I go into what I do, this is me, and this is the university I used to work on, and I used to do gene therapy, but I want to go in genetics, okay? So in science, and what's the big question here? What does genetics answer? What genetics is? It would say, the main answer is, who am I? We all want to know, who am I, and what does the genes do for us? For example, here right now, what I'm showing you is the evolutionary picture, where when the human became, or we are thinking what we are all about. So what are genes? Genes are the blueprints of the individual. Genes are what make us who we are. We all know, like you know, in your everyday life, we say, okay, there is a gene pool. We get some genes from our mom, we get some from our dad, and we become a unique organism, right? You'll agree with me, right?
Yes.
Perfect. All right, so what does the evolution determine then? The evolution determines why our genes are the way they are, you know? So I would go briefly into genes, and then we will do the fun part. So what are genes? As I am assuming you know that they are contained on the chromosomes. Now this is a picture of chromosome. It's pretty cool. Not that this looks like that in our body, but this is how we see it under a microscope. It's always fluorescent. So this is chromosome. Genes are contained on this chromosome. And we know the chromosomes are found in all the cells. So what do the genes do? It's the genes that are going to make the protein that makes the trait. Trait meaning what we look like. For example, my hair color are black. Lindsay's hair is different, or our eye colors are different. So that all comes from the genes that are contained on the chromosome, and this is what a chromosome looks like. So now in genetics, how do we study genes? This is how we study it. You see this, you see all this colorful stuff over here. This is exactly what a photograph of our 23, we have 23 chromosomes in our body. You know that, right?
Yes.
Okay. If you don't know, we all have humans. I mean, every organism has different number of chromosomes. We have 23. We get one set of chromosomes from dad, one set from mom, and after all the cell division occurs, this is what this is. And on our map, or when we photograph, like I said, it's a fluorescent photograph. This is what exactly each chromosome set looks like. So when a geneticist looks at these chromosomes from anybody, even by looking at them, we can determine if this individual is going to have a genetic disease. But we can only determine for the disease for the genes that have been discovered. Okay, this is not complicated right here. I just wanted to show something. That what are the uses in everyday life, even what is the use of studying genetics? We can study the inheritance, right? We can. So what I want, if look over here, this is called, in English you will say it is a family tree. In genetics, it's called a pedigree. So if you come to me and I ask you, okay, you know, tell me about your mom, tell me about your dad, how many brothers, sisters you have, this is what we write as. You see the square, that's the dad, the circle is the mom, and we show that these two people got married, they have 4 children, out of which 3 are sons, 1 is a daughter, Then we show another family. Now, and then they have 2 kids. This is how a pedigree in genetics is made. Okay? Now, when we color any of the squares or the circle, this means that this individual has a gene that can produce a disease. Okay. So basically, what is the use in everyday life is, one, we can see how the genes are transmitted from parent to offspring to further, and we can make a family tree to determine if they would have what kind of genes in their family. So we go to what would be the common applications of genetics in everyday life. I'm not going to the high-fi stuff, but in everyday life, even vaccines, vaccines are, do you agree that vaccination is important in everyday life?
Yes.
Okay. And then secondly, I'm just giving a couple of examples. In medicine, for example, we can treat genetic diseases by making custom protein. Custom protein meaning we can do gene therapy. That is what I want to come after in a moment, because that is my subject. That's what I do. But in agriculture now, I'm pretty sure you hear the word GMO every day. Do you?
Yes.
So, okay. So GMO, the genetically modified organisms, right? And there are people who say they're bad, people that say they're good. Why, as a geneticist, I would say they're good. Look at in agriculture, we can make more crop. There are third world countries where they don't have enough food. So here in the US, we can genetically modify our food, rice, corn, anything, and we can produce in bulk. So in a way, we can, work towards a world hunger. We can make disease-resistant crops. What would that mean is we won't have to use pesticides. So these are some of the applications of genetics. Now the big question is, what do I do personally? Personally, for like 15 years, I have worked on gene therapy. What that means is so there are diseases for which we can only manage a disease, but I wanted to find a method for which I can put a good gene in the human. Like, I'm showing you a picture of this baby. You can see that, right?
Yes.
Now he has this baby has a disease called Fanconi anemia. It's not anemia, it's not leukemia. It is a genetic disorder. And it was named after whoever discovered it, which was Dr. Fanconi. And so the reason you can see that this kid has weird features. The face is different. Look at the hands. This particular one has a missing finger. So even if we manage their disease, I wanted to find a way that I can put the good gene in this child so that that gene can make the protein that's missing, and then this child can grow up and have a normal life. Because a lot of these children, or at least the disease I study, they only live up to the teen years. So this is a close-up of, like, if you look at this hand, the finger is missing, here there are extra fingers, and so forth. So that is the whole idea of studying genetics, not the whole idea, my idea in medicine, that I can treat a disease. So now we have identified a lot of diseases, but I wanted to show this cartoon kind of thing Before we had genetics, for example, here this patient comes to the doctor, but the doctor is like, oh my God, I don't know how to treat you because nobody has your disease. I don't know what it is. So coming back to the patient I showed you, because the main symptom is that they die of bone marrow failure. They have a whole plethora of other symptoms, but the main thing why they cannot live beyond their teenage years, 16 is like a cutoff point, that they need bone marrow transplant. Now, what's the problem with the bone marrow transplantation? We need a matched donor, right?
Yes.
So not everybody in the world will find a bone marrow matched donor. So this means the method that we right now have is available for 25% patients. How does the bone marrow transplantation work? I'm showing you what I do with my little patients, which are the mice. Okay. So this is a healthy donor. It's a mouse. And what I do is, in the mouse or even in the human, we take the bone marrow out of the hind leg because that's the largest bone in the body. You take that, you count the cells, and then you inject into the sick mouse. The other thing with my work is, you see this mouse where I'm showing that this is the mouse is the recipient. I made this mouse. I genetically modified its genes so it is a patient. It has the gene missing, which is missing in the human patients. It is not making the protein, so it is sick. So I took this normal donor and I put the matched bone marrow transplant, and then I count the cells and I see whether he's becoming better. But like I said, this method is only available for about 25%. Then secondly, you hear a lot of things. Why does the bone marrow work? It's the stem cells. You hear a lot about stem cells lately, right?
Have you heard about stem cells?
Isn't stem cells like what a plant has a cell in the stem?
They're not sure what stem cells are yet.
Okay. So this is what I'm going to go through here. So why the bone marrow works? Bone marrow has, let's say you could call them stem cells as they're immortal. If stem cells is like a cell in the body, which is like a baby who has not yet decided what it wants to be when it grows up. That's a stem cell. And that's why they are so important, because they can become anything they want when they mature. And they are kind of immortal. The bone marrow makes them constantly. The second thing is that the stem cells would give rise to all other cells in the body, like red blood cells, white blood cells, and all the cells in the body have a lifetime. For example, red blood cells, they only live for 120 days, which is about 4 months, right? So every 4 months, our bodies get new red blood cells, which we never come to know because the body takes care of it. The bone in itself, the bone marrow knows 4 months are coming up, it's going to make new stem cells, and it's going to mature the stem cells, and they will make our cells. So, but the one thing about the stem cells is that they have a signal on them called a homing signal. What that means is, wherever you put a stem cell in the body, it is going to go back to bone marrow. You can put it in the leg, you can put it in the head, you can put it anywhere, it goes back. So, to do the gene therapy, this is where the problem sets in. We need to manipulate the stem cells and we need to manipulate the gene so that we can make the good protein and heal the patient or treat the disease. And this is called gene therapy. So what is gene therapy? It is the use of genes to treat a disease. In principle, the way I said, it's very simple. The principle is simple. Here's a healthy gene, we're going to put in the body, it's going to make a normal protein, and the patient is cured. But in practice, it's very difficult. Why is it difficult? We cannot just take a gene meaning DNA, right? DNA dissolves in water. You can have it in a little tube. So we cannot just take the tube, inject DNA in somebody, and say, well, it's going to go start making a protein. It won't. It's going to get out of the body. The other two problems with gene therapy is when we really do put the gene in the right way, we need to put it so that the protein is produced only when it's needed. Otherwise, we're going to get cancer, right? What's cancer?
Cancer is something that moves the body.
Exactly. So in cancer, the cells are growing uncontrollably. So if we put the gene in, we don't want to put the gene in so that it starts making extra protein. Then we're going to have another disease. And the second thing is, like we just discussed about cancer, so on a gene, on our chromosome, there are genes for different diseases. And there are some genes called oncogenes, which produce cancer. All of us don't have cancer because mostly they are dormant, they are sleeping. So with gene therapy, that's one of the challenges. If you put it in the body, it can wake up a sleeping gene. Then we get into more issues. But coming back to what I have done and how this can be avoided, what did the geneticists do? We thought, you know, how we all get flu. We get an infection. We get a bacteria or a virus. So how do we get that disease? How do we get a viral disease?
Bacteria infections.
They are going to put their DNA in our body, which causes the disease. So what we decided to do is take the virus, take its DNA out, because that's the DNA that causes the disease. So now we have to say like an empty shell. So we can take those empty shells and we can put the DNA that we need in that virus. Does that make sense?
Yes.
So what I said is we have any virus, like the influenza virus, the virus that causes flu, right? What we're going to do is we take its DNA out because DNA is what causes that infection in us. So we have just the outer part of that virus. So what we're going to do is we're going to take our good gene that we want to go to the patient. We're going to put our good gene into this vector. We called it a vector because it's carrying. It's like a cargo truck. Okay? So we're going to load this virus, which has nothing in it, with the DNA we need. And now this vector, we will put it into the patient where it will unload. It will put the right gene into that patient so that DNA will start producing a normal, healthy protein that we need. So that is the concept of gene therapy.
And I'm sorry, you do this with genes that are either missing or messed up in a patient? Is that right?
Yes, either they are, so only the genes that we have identified. For example, I took the example of Fanconi anemia where there is a mutation in the gene or the gene is missing.
Right.
So we will add a gene.
Okay.
Yes.
Can I explain that real quick to them again? Okay, so she's talking about, so you get a virus, like you get the flu, and it's a virus that has its own DNA. So they come and the virus gets into your body and it injects its own DNA in your body, and that's what causes you to get sick with the flu. So what they do with gene therapy is they take a virus, they take out that virus's DNA, and they put good DNA into it and kind of use it like she was saying, a cargo truck. So they use that capsule of the virus, they put good DNA in it, and they inject that virus with the good DNA into your body, and it kind of helps to repair where genes are either mutated or they've been messed up, or if the gene was missing completely. And it helps them to heal their patients.
Perfect.
Really cool. Yeah.
Yeah. So
Okay.
this is a cartoon. I'm going to show how I made it. For example, I just wrote transpose. These are the names, just forget the name. But the green part you see here, so I'm showing a gene where this green thing is what we need to go in the body. So here's the genomic DNA, the yellow one, right? And wherever I show this lightning kind of thing is where this green part is missing. And we want this green in this genomic DNA. So with the method that she just described, and here the DNA will integrate. So you see we got our normal DNA where there was a problem, but we did get our green thing in there. This means it's going to start producing the normal gene now, or the normal protein. So this replaces the sick gene with the new gene. That is the idea. So here I just wanted to show you how I did it with the mouse. Like I said, you take the mouse, harvest the bone marrow, and this is the way it would look like. So you take the bone marrow or the stem cells, and it all happens in the lab in a petri plate. So here is this virus we talked about. We're gonna mix them in a petri plate, and from there now we have these cells which are carrying the healthy DNA, because we have mixed the sick cells with a virus that has normal DNA in it. And this will go back into the sick mouse that I told you I made. And you can it's just an injection like an IV. Like for the patient, you do a bone marrow, and then you just start counting. You take the blood every week or a month, and you can see whether the new gene is being formed. So I don't want to go into that methods, because they are like you isolate the DNA, then you can look on a gel what it's showing. This is the method. Just the same, you harvest the bone marrow, you mix the cells with the required gene, because there are different methods. You can even do a little electric shock to the cells, and then you put them back in the patient or the mouse. So in nutshell, what I wanted to say, that the genetic diseases, if we study them, they are a window into DNA repair. So there are a lot of functions. You can study the genetics, we can study inheritance, we can study population genetics, we can apply it to correct a gene or, cure a disease, or you can go towards science and you can study the DNA or how it's repaired. So, similarly, for cancer, we can have the same, we can study the cancer. How did they study the cancer? It came from the genes, and from there we can study the cancer biology or how to prevent it and how to treat it. This is one of my publications, but that's the work I wanted to discuss with you.
Do you have time for some questions, maybe?
Yes, of course.
How do you transfer the genes that have like if a mouse has has a bone disease, like, do you replace it with another bone?
He's asking, like, if a mouse has a bone disease, would you replace the bone, or how would you go about fixing that?
So that's different. Let's say the bone disease is because of a gene, right? Then it would be the same method. We need to know what gene is causing the disease in the bone. So we would isolate that gene in the lab. We are going to do the same method. We are now going to take the same virus or a bacteria. We're going to put the good gene in, and then we're going to put it in that mouse or the patient. And if that's the gene that's causing it, it should correct it, yes.
Keep in mind that she's talking about genetics. So we're not talking about things on a large scale as far as like replacing a whole bone or replacing a whole organ. We're looking at it genetically to look at the DNA to figure out how can we fix the problem so that it's not causing that disease in the patient. Does that make sense? Okay.
I mean, lately, according to genetics, I mean, so liver is it's the most easiest. We can grow it in the lab. And this is what they're trying to do. Liver is the only organ in our body that can grow. So if somebody needs a liver transplant, you can just give them a chunk of liver, like a tiny piece. It will grow into according to the weight of the human. So coming to saying what you were asking, other than a genetic disease, we are trying to grow kidney, for example, in the lab, and maybe those artificial kidney can be transplanted, or even the bone. We can have the cells, but we have not succeeded in doing all that.
How can you replace a piece of liver instead of bone?
He's asking, how can you replace a piece of a liver and it would grow into a full liver?
That's called a liver transplant. So let's say somebody got cirrhosis. Maybe somebody drank too much alcohol, and now their liver has failed. So their liver would be taken out. It still has to be a matched donor. That's the whole thing. That's the reason gene therapy comes in. Then we do not need matched donors. But in this case now, we will match a liver, and then we can just take a little chunk of the donor liver and put it in the person whose liver has failed. We will take that liver out, and that's the transplantation that happens.
Through mitosis, cell division, it divides and it grows.
So, like, they take the bad liver and take a chunk out of the good liver and, like, do they combine them together?
No. What I'm saying is bad liver has to be taken out of the body.
Oh.
A new liver needs to be put in the body. But for the lung and stuff, you need to see, you cannot take a baby lung and put it into an adult. Liver is different because our liver grows according to our weight of the body. So like for a baby, you can take a chunk from an adult and put it in the baby. It will shrink.
So she's saying, for example, so the liver, if someone, like she mentioned, when people drink a lot of alcohol, a lot of times they have liver failure and they have to have liver transplants. So they take the entire bad liver out. And she's saying that what they can do is take a piece of a liver, of a good liver, and implant that into the patient. And that liver will grow. We talked about mitosis and cell division. That liver will grow into a complete liver. You'd have a new organ for that person to survive with.
How long would it take to be, like, fully grown?
How long would it take for that piece to develop into the full organ?
That liver, it's usually taken pretty comparably, but it would take just a few weeks because, that's a transplant procedure anyways. They need to get radiation and all that stuff. So in a few weeks, that liver should get adjusted if it hasn't been rejected by the body.
Think about when we talked about fertilization and how you're starting off with 2 cells that combine, and within 9 months for a human, you have a full baby. So in 9 months, it went from these 2 cells combined for that zygote and developed into a whole baby within 9 months. That's really quick. So with this, you're just developing one organ. So a couple of weeks.
We're not developing like from the scratch. We are
Right,
putting
exactly.
it in.
Right.
So it's only growing up to the weight of the body. That's all. So let's say we put a huge liver in, it's going to shrink to a small liver. If we put a small liver in, it's going to grow to the size of the body.
Okay, so it adjusts to the size of the patient.
Yes.
Awesome.
But it does not happen with anything else. No other organ in our body grows on its own, like kidney or heart or anything else.
Right.
The gene disease, what you were talking about, do you catch it or were you born with it?
He's asking the one she was talking about, the one that she works with.
Yes.
The genetic disease that you're talking about that you work with specifically, is that something that people are born with, or do they catch that?
No. That's why it's called a
genetics. Yeah, so they haven't learned.
It's a genetic disease, meaning, so the disease I was talking about, it's an autosomal. We have autosome chromosomes, right? Autosomal recessive disorder, meaning both the parents will have one copy of defective gene. So the parents are not sick because they are each copy. There are two copies of the gene. One is defective. But when they have a baby, the way we would do with the pedigree I showed you, 1 in 4 of their children will definitely have a disease. Because 1 in 4 children will have both the copies, which would be defective.
So they're born with it?
Yes.
It's something we haven't talked about yet, but it gets passed on from the parents to the offspring, but it doesn't necessarily mean that the parents have that. They just, what we call carriers of that trait, and they pass it on to their offspring.
Genetic diseases are always, they are passed on. You don't catch it like a flu.
She's asking about the rarity of genetic diseases. You want to know about hers in particular, or just genetic diseases in general? Is it rare for people to catch or to inherit genetic diseases, I guess?
Well, the one I discussed is very rare. It's mostly for the Ashkenazi Jewish families because they intermarry, right? This is how a genetic disease would actually get concentrated if there are two individuals who are carrying a gene that is defective, then definitely their kids would have the disease. But if they're getting married outside different areas, then they're not carrying the disease and the gene gets diluted. However, like multiple sclerosis, we have the gene for it now, or we can go with Alzheimer's disease or Parkinson's disease. So those we hear, they are becoming pretty common, I guess. And that's one thing where we are actually actively trying to do gene therapy or the stem cell transplantations.
So some of them are more common than others.
Yes.
That thing at the beginning, what's that? OHS.
OHSU is the Oregon Health and Sciences University in Portland, Oregon. That is one of the major hospitals, and or it's a teaching hospital, and it's a research university. And this is where I spent like the past 15 years.
Is that where you are now?
No, now I started because of all this, especially. That's why I was bringing the issue of stem cells and GMOs and so forth. There is a lot of misinformation in public or about the vaccines, if you guys heard the news. So I have started a nonprofit organization, and I have a website where I have a whole team of MDs and PhDs, and we kind of write the articles based on the entire literature out there, so to educate the community. That's what I do now.
The Lou Gehrig's disease, that has like something going on with like the genes, the body.
Lou Gehrig disease I don't think its gene is identified, but it is a muscle wasting disease. and I do not think that we know what gene causes it. So unless we have identified the gene, we cannot do a gene therapy because we won't know what gene to manipulate.
Thank you so much. I think they enjoyed it. Yeah.
Thank you.
Did you guys enjoy it? Okay. Thank you. You guys are welcome to email me, or your teacher can send me the questions if you have any at any point. I would definitely answer.